Hi guys. I'm sure you must have learned a lot of things in this integrated systems biology workshop. We thought we'll end the sessions with a way of how to write reviews, especially the systematic and meta-analysis review. So that would end on a sweet note. Why? Because whatever you have learned, you might have to, whatever research one does, that is only half work done, and the other half would be to publish it, writing papers. So before you even start with research, whatever the kind of searching, the topics, etc., you do, almost covers under, falls under the review. How do you write a review? And review also, if I have to say, is not a very new thing for all of you. Like it is almost similar to that of a long answer essay, you write for during your masters and all, long answer questions, but that is the only similarity, that is where it ends, that similarity between a long answer and that of a review. Now, reviews is almost, as I told you, is like a summary of something. Now, when we say review, there are the different kinds of review, and something along the lines of an essay, which usually write, or the long answer, would be called as narrative review. That is, you're narrating something, you're topic, you're summarizing something on a given topic. Now, why should we go with systematic and meta-analysis, and how would this much more better and more relevant than a narrative review? That's what this talk will be on, and hands-on will be also on those lines. Now, the systematic and meta-analysis review, me and Indrani will be taking care. Indrani will take part, take into the 29th date, 29 July session also. Okay. So now, before we go, why should we do a meta-analysis or a systematic review? Let me tell you a story here. Now, this is a book called The Pocket Book of Baby and Child Care, written by Benjamin Spock. Okay, and in that particular book, there is a statement which is called, which says, I think it is preferable, to accustom a baby to sleeping on his stomach from the start if he is willing. He may change later when he leans to turn over. Now, this was a kind of statement which is included in a, a kind of childcare manual written by this particular gentleman. And this, book had, many editions and sold millions of copies during those 1990s. And if you observe, the advice was not unusual, in that many pediatricians made similar recommendations at the time. It's, it's like a hidden trial. You might have observed a baby sleeping on its, tummy. But there's a huge but. During the same periods from 1950s to the 1990s, more than 1 lakh babies died of sudden infant death syndrome, which is called as SIDS, also called as crib death in United States and cot death in United Kingdom, where a seemingly healthy baby goes to sleep and never wakes up. So, what is it then? So, in the early 90s, researchers became aware that the risk of SIDS decreased by at least 50% when babies were put to sleep on their backs rather than on their tummies. So, governments in various countries launched educational initiatives such as back to sleep campaigns in the UK and US, which led to an immediate and dramatic drop in the number of SIDS deaths. So, what is it, what that have to with the systematic review is that, there's a author who wrote that advice to put infants to sleep on the front for nearly half a century was contrary to the evidence available from 1970 that this was likely to be harmful. So, a systematic review of preventable risk factors for SIDS from 1970 would have led to earlier recognition of the risks of sleeping on the front. And it might have prevented, if someone had done a systematic review on the risk factors which would have prevented SIDS was done by, any of the scientists, many of the lives of the infants would have been saved. Is what he had summarized. Okay, so this is one that meta-analysis, systematic review analysis, where it was told that infant sleeping position has a lot to do with the sudden infant death syndrome. Okay, so this is a gentleman called Ian Chalmers, and he had written, had given a talk called "The Scandalous Failure of Scientists to Cumulate Scientifically." Now, what it means is that, so the theme of his talk was that we live in a world where the utility of almost any intervention will be tested repeatedly. Rather than looking at any study in isolation, we need to look at the body of evidence. So, while not all systematic reviews carry the urgency of SIDS, the logic of looking at the body of evidence rather than trying to understand studies in isolation is always compelling. Now, what he means here is in his talk, he said that, say, suppose I'm doing an experiment in my lab in my country, and a similar experiment has been replicated in different labs around the world. Now, are we going to get the similar outcome or a different outcome? Who's going to tell us? So, a person who's writing a review, systematic review will be the person who will be doing that. That is, the same experiment carried out by different people across the country, if we summarize and look at the consolidated or cumulative effect or of the data, now you will see where the similar pattern is seen there, or is it different? If it is different, why? So the question of why, which starts with the scientific research, has to be answered periodically through systematic reviews. This is one of the best example or one of the uses of systematic reviews. Okay, so whenever starts whenever someone does a research, we should know, ask, why did you start that particular research? There should have been a solid question, right? So what did you do to answer that question, and what answer did you get, and what does it mean anyway? The same four questions are also answered even in a systematic review. However, we try to pull up studies from different scientists who have done it all over the world, and try to see if it is replicable, or the results are replicable or not. Now, if you can see, this is a small public summary, wherein there are they are addressing random clinical trials of, say, a question, okay, which was done in 1997, 2001, and 2005. Now, if you see, till 1997 there were 26 studies. So what was the first question? First trial addressing the question. Okay, there should be some question. Is this drug, useful for treating a disease? Might be the question. So if you see, this is the question. Now, if you see the numbers here, we'll see how many of these studies have addressed all these four questions. First trial addressing the question, only one study has done it. Then, contained an updated systematic review integrated the new results. That is, if you have done a research, similar ones, if anybody has done, you should be able to compile it and then present it. Then, discussed a previous review but did not attempt to integrate new results. Did you see? There were only four such studies out of this 26. Next, no apparent systematic attempt to start news to set new results in context of other trials. And if you see, the majority is 19, which is, it's a very bad number here. They should be able to fall under this category. That is, your results with that of the previous results, summarize it and then present. Similarly, if you see the trend of the number here is very high. That is, but of course in 2005 at least they've made the number is comparatively less when compared to the other 2 years, where only there has been at least they've tried to put in some results. Now, why is it important? Okay, now why is it important that we have to have a systematic review? The second use is, remember, re- clinical trials are highly ethical issue. You can't just use humans as guinea pigs, and for that matter animals also, as because we are talking about lives here. So it's very, very important that before you or before one scientist talks about a clinical trial, he should have a proper knowledge. If the same experiment done by 10 different people didn't show any result, the 11th experiment, the probability of the 11th experiment also not showing the result will be very, very high. So you're not going to waste again, the lives of, people here, or for that matter, the lives of animals here. So it is a systematic review is like a pause before you proceed to the next, research. So he, Ian Chalmers was, was of, concluded that reports of clinical trials especially should begin and end with up-to-date systematic reviews of other relevant evidence. Okay, so maybe we should all have, this kind of mentality there whenever we think of doing research. So reports of new research should make clear what contribution the new evidence has made to the totality of the relevant evidence. Say suppose you want to again conduct a trial, clinical trial, you should be able to make sure that you are adding some value to that clinical trial. Okay, 11, 10 studies have proved wrong. In the 11th study, I'm making this measurement, these changes, and I'm hoping this would be a better trial will with giving better results than the rest of the previous trial. This should be the kind of research work based on relevant evidence which should be done. So in short, people have suffered and resources have been wasted because new evidence has not been set in the context of up to date systematic reviews of all other relevant evidence. Okay, so this is very important. Now that brings us to the next question. What is systematic and meta-analysis? Now these both are almost interrelated. Now we'll see meta-analysis refers to the statistical synthesis of results from a series of studies. While the statistical procedures used in a meta-analysis can be applied to any data set, the synthesis will be meaningful only if the studies have been collected systematically. So you see, a meta-analysis base is always a systematic review analysis. So, a systematic review gathers all information with respect to a specific concept or question inquired using systematic methods approach. Now, what is the systematic methods approach? That is what this talk is all about. Okay, so once you have gathered all the evidences, information you want through a systematic methods approach, you apply in the final, step a statistical measure which makes this systematic analysis a meta-analysis. Now, if you see, on the whole as a gist, the systematic analysis has these all steps. Planning, that is, it starts with researching for existing reviews or protocol, formulating the review question, developing a review protocol, search strategy, and then systematically search databases and download citation to a reference manager, select the study, which kind of studies you would like to include and exclude, extract the data, what is it you're exactly looking for, then synthesize the data, review, writing the review, and publishing the review. If you see, there will be extra steps here after the data, some statistical analysis for the meta-analysis. Now, let's start with searching for existing reviews and protocols, which is the first preliminary search. It is important to conduct a preliminary search to see if others have done it. Okay, if others have done it, just by yesterday, there are latest publication on whatever topic you want to write a review on, or a meta-analysis or a systematic review on, it will be waste of your time because there's no point in rewriting the same. Okay, so always start with search. Now, where can you search? Now, you have something called Cochrane reviews. Cochrane reviews. Sorry. Yeah, Cochrane reviews, and then which has a database with called Cochrane Library, and then Campbell Collaboration, and an international social science research network. So social science, when we say, it will have a kind of survey kind of studies, etc. And then Sinahall. Now this is a database providing full text of nursing and allied health journals. And then Doffer, Jones Bridge Institute EBP database. Medline is basically our related to National Institute of Health, NIH. So you'll have PubMed also covering in it. Then Prospero. So these are all the databases links. We'll be providing these this particular slides. You can click on each of these links and see. Okay. So you start with searching in all the possible database. You can even search in Web of Science, Scopus, Dimensionality, etc. Also to search for all the possible studies which has been done, Google Scholar, etc. Now, once you have done, you can look for registered protocols. Now, what happens is whenever you want to do a systematic review, there are certain kind of registries where you can register saying that I am going to work on a systematic review on this particular topic. So what will happen is you will be given or assigned a number who which you can use it while writing the paper also after you're finished. So these are the registered protocols where you start with. Okay. Then after that, how do you develop a protocol? So now, what is a systematic protocol, review protocol? Now it has all these steps. Okay. It has an objective. What exactly are you trying to question? Which is a question you're trying to answer? A review question, a score of the review, the research group, search strategy, methods, inclusion exclusion criteria, quality assessment, procedures for data extraction and data synthesis, and a project timeline, etc. So you can find these examples of protocols if you click on them for your later reference. And then how would you go about starting a systematic review? Usually you might have heard about this term prisma statement, which usually helps us in the helping you guide through step by step. Okay. So usually whenever you write a systematic or meta-analysis review and you try to submit to a paper, they'll say, have you checklisted with the prisma guidelines? Okay. So it would look like this. So identify the report a systematic review or meta-analysis or both. It can be both also. Does it have a structured summary? Does it have a rationale? Does it have objects which will with reference to participants, interventions, comparisons, outcomes, and study designs? What is the protocol you have followed? Okay, how many, like which all databases have you accessed? If available, if you have registration information including registration number, then the eligibility criteria, based on what are you having this exclusion inclusion criteria. Then information sources, then present full electronic search strategy for at least one database, including any limits used such that it could be repeated. That means if suppose you have done it and someone else want to exactly try to replicate your study, they should be able to do that. Okay, so all that information needs to be given. Then data collection process, how did you do it? How did you how many total number of articles were selected from which? How did you filter out to come up with only a few selected numbers? And then list and define all variables from which data was sought and any assumptions and simplifications made. For example, you would like to say, I want to see risk of say what do you say, vitamin D levels with for the causation of say in the in the associated with bone disorder or skeletal disorder or say specific rheumatoid arthritis, osteoporosis. Now what would you do? You will try to search for all those studies which has done work on um, levels of vitamin D in osteoporosis, for example. So what would you say? You might get a good number of articles. Now what happens is there is a given range which will be differing from one country to country or lab to lab. So you may have to normalize it. Okay, so that is what here methods for any simplifications made. So you are coming to consensus with the kind of work you want to show. Then risk of bias in individual studies, describe methods used for assessing risk of bias of individual studies and how this information is used to in data synthesis. That means, have you used any bias? I am including these, or I'm excluding those. Did you have some bias? Why are you showing that bias? Why you've excluded things? That all needs to be done. Then a summary measures, that is risk ratio, odds ratio. If you have meta analysis, you will have forest plots and effect size calculations, etc. Then once all this is done, you have to tick mark on this, and you have to report in your review which page has all these information. Then now, as I told you, after your search, now you want to formulate a research question. Okay, I'll tell you one of the most common formula kind of thing. See, if you see, P is for people or population or problem, I or E for intervention, exposure or event, C for comparison or control, O outcome, T is time if applicable. So you have PICO or PICOT or PECOT. These are the kind of formula. Okay, so the fewer elements you use, the broader the formulation of the question. For example, what do you mean by broader questions? For example, I'm asking a question here saying, what type of programs can build mental resilience among student nurses? So if you see, there are a number of factors, variety of factors, which, matters for person to person, which changes from person to person. So this makes a broader question, examples of a broader question. Now, what do you mean by narrow question? Narrow question is you try to put or fulfill each of the category of the PICO formula there. For example, if you read this particular statement, in patients undergoing a surgical biopsy, so P becomes, what is P here? P is for people, population, problem. So what is the problem here? Patients or the persons. What is P for people also? So who are the people? You're not considering everybody. You are very specifically picking patients undergoing a surgical biopsy. Now, how does mindful meditation? This is the intervention or the exposure or the event based on which you would like to see. Does this mindful meditation compare to hypnosis? Now, C is for comparison. You're comparing two different kind of the psychological events on patients undergoing a surgical biopsy and effect of the use of painkiller. They would like to see the outcome here in the first 24 hours after the biopsy. Now, you see, PICOT, all the criteria have been met here. Okay, so this would be almost basically the title also of this particular systematic review. Now, for example, another second example where I would like to narrow down the question. Now, if you click, pediatric diabetes in PubMed, you'll see that around 21,000 results are present. Now, I can't write, you can't read. It's impossible to read all the 21,000 kind of, articles into 10, say I'm going to summarize it. Okay. Now, this is a broad question. And the broad question doesn't, can have different answers or different questions. In pediatrics, what am I looking at? Am I looking what causes? Am I looking at what risk factors might be? Am I looking at the management, or am I looking at the diabetes? So many questions have come. So we'll narrow down this. Now, how do I narrow down? Because you'll have articles related to comorbidities, for example, cardiovascular medication use and cardiovascular disease in children and adolescents with type 1 diabetes. This also comes under pediatric diabetes. Next, drug therapy, pharmacotherapy options for pediatric diabetes. Now, there can be number of different drugs used, which can be used. Still a broad question. Or you can use, as I told you, social determinants of health, like what causes pediatric diabetes. Still, it's a very common, broad question. Now you have to narrow down. Okay, so look at this question. What is the efficacy of metformin versus rosiglitazone on glycemic control for children with type 2 diabetes. Now, if you see, it is covering T. Who are the children? We are like, on whom are we looking for this systematic review? Children with type 2 diabetes. Mind you, there is type 1 diabetes also. Some people can, some children might have even um, uh, what do you call that, neonatal diabetes also, right from the day one they have diabetes. Now I'm only looking at in children with type 2 diabetes. Now what is the intervention? Efficacy of metformin versus rosiglitazone. Versus, so I'm comparing metformin with rosiglitazone to see what glycemic control is the expected outcome. Here if you see, there is no timeline. Okay, so yes, you can, sometimes it happens that not every kind of study you're trying to look into, timeline has to follow. uh, Everybody listening here, am I fast, am I slow, is it okay?
Yeah, we are listening.
It's perfect. Okay, thank you. I don't know, because sometimes in a online session, I don't know whether people are sleeping or am I talking to myself. Yeah, thank you. So as I told you, there are registries available for a systematic and meta-analysis. If you go to this website called Prosero, what happens is you have to register and say, this is the title on which I am going to write a systematic and meta-analysis review by so and so time. So what will happen is they'll assign you this number, Prosero number. You can use this number or include this number also and modify, and write it in your methodology session that this particular review or systematic review has been registered under Prosero with the so and so assigned number. Okay. Okay, first talk about literature where I didn't sleep. I didn't understand. I didn't get you, Rakesh. Literature, we'll come to that. Why? Because that is the kind of, hands-on for you people. Let me go through this complete step because there are a lot of steps involved here. So your homework would be a lot, to do. Each of you might be from, interested in different kinds. Some might be interested in plants, some might be interested in animal research, human also. There's cancer, so many diseases. So each one of you will assign a task wherein, or you have to check and tell whether all these procedure or any steps have been covered or not. So this will be the first assignment. Okay, we'll move forward. So apart from Procero, you have Cochrane reviews also, where you can register your title with even this particular database. And there are others. Now, once you have decided, see here till here, what happened? You have come to the stage of defining your question. Okay. Uh, Defining your question here. Now, once you have question, have a question, very clear cut question, you go and register your question, okay, in one of these registries. Now you have done that. Now the actual work starts. Like till here, only 1/4 work of your systematic review has been done. The next part would be, now, of the many searches you have given, you have to set an inclusion and exclusion criteria. Now, why? Because that will help your research focus, even more. You narrow down and, your question becomes very tight here. And the selected studies will be able to answer your question. And the selection bias will be minimized. Okay. So that is the reason why your inclusion exclusion criteria, and based on this inclusion exclusion criteria, you can modify your title at the later stage also. There's no problem in that. Now, inclusion exclusion criteria are most commonly determined after setting the research question and before conducting the research. However, as I told you, you can reconsider it and change the title or your inclusion exclusion criteria according to the question you have got. So these are some of the common inclusion criteria. Say suppose you have too much data, like 1 lakh, 2 lakh search results is coming, so it is very impossible. Even after you put in filters also, you'll get thousands of papers, and that will be really like out of person's scope to finish writing a systematic review. So what you can do is you can narrow it down to at least 1 decade or 2 decades, etc. That would be one inclusion criteria, exclusion or inclusion criteria. Then you can also very specifically put it in categorize into data which includes in a specific geographical region. Like for example, you're looking in some cancer data or cancer drug trials, etc. What happens, you can only minimize it to Indian studies. Okay, so that will make you it's a very specific topic. Then you can even limited to a particular age or gender or the location of the participants, and then review limited to randomized control trials. Even randomized control trials, if there are too many, you can ask very specific question. No, I want to only look into this particular topic. So that is how I can reduce, or the number of papers I'm analyzing, and making the question very very tight. Okay, and then you can even say that identify database and search strategy. Some reviews may include gray literature, like it's neither that nor that, there's somewhere in between. So you can say I'm excluding this because this doesn't fall under the criteria which I am looking for yes or no answers. Then you can also type types of interventions using products having only natural ingredients in them, or the natural, say for example you're talking about anti-cancerous agents, we'll say we are not looking into any kind of anti-cancer drugs which are synthesized chemically, artificially. Rather we are looking for only those which have natural ingredients. Okay, so something like this, the inclusion exclusion criteria can be done. Next, now inclusion criteria is usually reported in the methodology section. Okay, after your search, which databases you have searched for, which keywords you have used for, then you can say that I am including only these studies which follows the inclusion criteria of this and exclusion criteria of this. Okay, then as I told you, after you finish, you keep on having to keep checking back your Prisma guidelines. Most of the time it is Prisma. Apart from that, based on the type of systematic review, most of the ones mentioned here belong to humans, or to some extent animals also. So if suppose you're doing your systematic review on plants, say for example, you will have to check for any other guidelines, specific guidelines which are exclusively applicable for plant-based research. Depending on that, you may have to check those lists and keep always having a look at that. That will make your meta-analysis even more good, excellent, to the level of excellency. Okay, and it also helps to reduce the risk of biases. Okay, so for example, here you have Cyril's ROC tool, which is by the Systematic Review Center for Laboratory Animal Experimentation. So there are certain guidelines given exclusively for systematic review of animals, animal studies. Similarly, you have other useful guides on which you can just have a look at it. Okay, healthcare interventions, health evidence. Most of the time, systematic reviews, almost like 90% of them belong to human healthcare, etc. Now, as I told you, PICO was one of PICO or PICOT or PECOS. These are one of the frameworks. Apart from that, you have also have frameworks like PIC. That means population, phenomena of interest, and context. Spider or sample, phenomenon of interest, design, evaluation, research type. Spice, setting, population of perspective, intervention, comparison, or and evaluation. So there are different kinds of frameworks. So it's not always PICO, but you can have other frameworks also. But having a framework is very, very important, because when you write a systematic review and submit to a journal, what happens is they'll ask you which framework does this study fall under, which framework have you used to analyze your results. Okay, so and then you have to keep recording your systematic review process. Why? Because the same thing, checklist, guidelines, frameworks, and keep a proper transparent and it should be replicable. Till here, whatever steps you are doing should be replicable. So that is one of the main criteria why recording all these, because sometimes people ask how did you do. please upload that particular Excel or backend data, whatever you've done. And typically, if you look into any systematic review or an meta-analysis papers, you'll see this flow chart. Okay, flow chart which starts with records identified, which how many databases, how many registers, then any duplicates removed. Because if you are searching in 2-3 databases, for example, you are doing the same search from PubMed, from Scopus, from Web of Science, what happens, there may be a lot of duplications will be there. So you have to say total number was this, what are the duplicates removed, and how many are you marking ineligible or why, etc. All these information with the number have to be properly jotted down. So finally you may start with say 1 lakh, but at the end of your included studies in the review, you might hardly get to sometimes 20, 50, or even as less as 5 to 7 also. But yes, even doing that is extremely important and time taking, so it's not going to be waste. So you even with 5 to 7 such studies also, you can write an excellent review. Okay, provided there is no such previous reviews on the set topic. And then again, you have the question is really really good, so that even 5 to 7 studies are enough to start to have the acceptance of this systematic and meta-analysis. Okay, so you have on these if you click on these links, you have examples of recording sheets, how to draw a diagram for prisma flow chart, etc. So these can be used. So I've search even for there are systematic reviews used in animal research, so you can click on these where there are a set of guidelines and frameworks for those working on animal data. As for the plant research is there, yes, there are also systematic reviews and meta-analysis in ecology and evolutionary biology, for forest restoration, plantations, etc. And so you can try even here also. For example, what kind of treatment is good for the given crop, or is there which genetic engineering procedure is very good for what do we say, gene transfer in a particular plant species, etc. So such specific, you have to be really really clear about the kind of question you're asking, which parameter are you searching for. So based on that, the meta-analysis can be done even in agricultural research also. So this is one of the ICMR publication which is openly accessible, a beginner's guide for systematic reviews. This is mostly, since it's ICMR, it's mostly on human health only, by the government of India, which has a detailed steps involved, how to do, what to include, what are the same kind of sites, which sites has which kind of information, etc. So you can as well look into this while you're doing it. Now that was the story of systematic analysis. Now if you want to do meta-analysis, as I told you, systematic analysis is the base of a meta-analysis. So what you do is, after your screening of inclusion and you are come up with the limited number of titles for to them to be included in the systematic review, what you do, you retrieve the PDFs, that is the original articles, and then you look into the data. Okay, so systematic review, what happens still here, if you are able to properly kind of summarize what has been given in this 10-12 papers, that will be the end of systematic review. However, if you add some kind of statistical data here from these studies, for example, you might have seen meta-analysis on gene mutation, okay, in particular disease. Okay, what happens is here you select or filter out as many studies as possible who have done the similar kind of mutational analysis which is associated with a disease. Now what you do, you'll see title number 1 or research study number 1, how many samples have been analyzed. especially among the disease. Say for the first research study, there are around 100 diseased individuals, and like 99% or almost all are getting the disease. Say, so you mark it 100 numbers, and this is the first study, and it is showing yes, it is associated. Now you mark this table similarly with all other studies. Say now you cumulative, you add up all those things, and if you say similar kind of results, then you are cumulative, you're adding up all those studies which are saying positive associated with disease risk, and say yes, this particular mutation is very much relevant to the causation of this disease as replicated or as seen in so many studies. Okay, so this is what a meta-analysis will do. So what happens is, for meta-analysis extra, you might have to use certain statistical tests, like the forest plot or the violin plot, and you use RStudio. Apart from RStudio, you can even do it in SPSS and other tools also. Now, if you see, there's something called fixed effect model, and random effect model is what you will be seeing in a meta-analysis. In simple words, what it is, fixed effect model is, you are showing that all the observation among the different studies is caused by sampling errors, or almost similar. And whereas in random effect model, different studies exhibit substantial diversity. Say for the same, for example, if it's not a mutation, there is another SMP which is seen both in controls as well as patients. Now what will happen, you can't say that it is affected mostly in disease, it is also seen in control. So different effects will be there. Some might say yes, some might say no. It's like a mixed data will be there.
Manjari, 1 minute. There is a
Yes, yes.
fluctuation in your voice.
Oh, I'm so
Sometimes it
sorry.
is low, sometimes it is high. I
Am I
think
saying no?
now it is low.
Oh,
Low means it is like somewhere inside a well kind of difference is there.
okay. Right now?
Now it is good.
Okay. So, we have for this fixed effect models and random effect models, which are kind of statistical done, tests done on the data which has been taken from different sources. Okay. Now, this part we can discuss it in the next session of 29th, which will be taken up by Hindrani. I mean, more detailedly. So, there are different tutorials where you can have, step by step they are being shown that how to do meta-analysis in IBM SPSS, and then how to do it in Stata. And we already had a workshop on this meta-analysis, how to do it in R by Shinduja. For those who are interested, you can start watching this after collection of data. Again, we'll be talking about the part of this, the meta-analysis part, on the session on 29th. Okay. So if you see, there's especially in human studies, you have simple, the weakest to strongest evidence. You have case reports, opinions, and letters, which will be considered the lowest among them. Then you have animal trials and in vitro studies, cross sectional studies, case control studies, cohorts, RCTs. So meta-analysis and systematic reviews have highest place among the scientific evidence. So in summary, you can see the difference is simple between systematic and review is that you do an extra statistical test on the information which has been accumulated or collected in a systematic review. Yes.
Then your voice is varying, means sometimes it comes clear and sometimes gets slow, too slow that we are unable to
I
hear it
don't know. I've
properly.
been
There's
yeah.
variation in your voice.
Okay. I mean, I'm almost at the end of the slides of the talk here. Now, I audible.
You are audible.
Hello.
Eh? Always you are audible. There is no issues in listening you. But there is
Okay.
fluctuation in your voice. We couldn't maintain the sound. Sometimes it is too loud, sometimes it is too low to hear. It is like that.
Okay. Okay. Yeah. Yeah.
Maybe the
But at least
distance.
I hope I've been able to, convey what I wanted to tell. If
Yes,
I'm,
yes, there is
if
no
I'm off,
issues.
yeah, yeah. I don't know. I've been, putting that mic near my mouth only, but I don't know. So this is the starting of the, what do you say, uh, the study which we are, I mean, systematic analysis and meta-analysis. So you have to go through all these set steps first before we can get into meta-analysis. So this week's hands-on is all about that only. So before that, let me answer the questions. Put in the chat box, and then maybe I'll we can have this discussion on what will be in the hands-on. Okay. Okay. Zayul is asking, ma'am, please assign exercise so that we can write at least one short review, or if it is possible. Yeah, that's the whole plan. I wanted to, make sure that at least one or two proper systematic analysis comes out of this integrated workshop. So we'll do that. So before that, so this week's assignment for you all is, each one of you, have some interest. Like for example, legume lab. What, ma'am? I forgot. I keep forgetting your name. Please.
Yasin.
Yeah, Yasin. You work on plants, right? If I'm not wrong.
Yeah, I work on plants. Legumes I work.
Yeah. So yeah. So each one of you, depending on your interest, there will be two assignments here. First one, depending on your interest, pick 10 meta-analysis or systematic review analysis papers and see what is the kind of framework they are following. Okay, so what will happen is you have to look into the protocol here, methodology. How did they go about? Which databases they have searched? Then, what is the exclusion inclusion criteria? Then, what is the research question? What is the framework? So that needs to be written down properly again. Okay, so 10 such papers, it can be animal work, it can be humans. Again, in humans you have different kinds of diseases, so you'll be working either on cancers or diabetes or xyz. It's up to your interest. Okay, so 10 papers, each one of you have to pick up 10 papers, already published 10 papers, and see which databases have been, all the steps have been put in. So I'll share all these slides with you, which has links to, various kinds of databases or all the questions in there. So that should make your life easy. So you have to do that, number one. Number two, now that you have seen papers in your subject area of interest, you have to at least form a question and a framework till there, and try to, search. It's all about searching databases. Search, you can use dimensionality, publons, PubMed, Google Scholar, and those who have access for, Web of Science and Scopus, you can do that better. You can help each other. Okay, so try to come down to a simple, very narrow question which is not there already. I should not see already a repetition of some others XYZs. Okay, so come up with such a title. Based on that, we can move forward. Can I get a yes or no's here? Okay, before all that, any questions or doubts here? Please. Kindly unmute yourself and let me know any questions.
It's a very good lecture, provided we get some kind of assistance in citation management.
Mm-hmm. I
Uh,
didn't get you.
like reference management, some tools for
Mhm.
reference management in while writing papers to add or delete a citation and automatically format it for a particular journal. So that
Yeah,
is
it's basically, yes. It's almost like a bibliometric analysis to start with, but you are, formulating a question out of all the research done. Um,
no, I couldn't. I think I have not conveyed my point.
yeah, I didn't understand. Sorry.
And something like EndNote kind of tool we are asking. Any latest?
No, actually, it is not needed, right? First of all, it is all about the search of the titles. It's mostly on the Titles you'll be looking at, even an abstract reading also, which should have you rough idea on what you want to search for, what is the question you want to formulate on.
No, 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 no. I'm asking something extra, not from your lecture. I'm
Acha,
saying,
acha, okay.
I'm saying if
I think
something,
Zotero is there, which can be used, if I'm not wrong. That's a free to use. Yeah, Mendeley is there. Zotero is there.
I never
I
used.
can post. I can post such reference ID. Of course, EndNote, but EndNote, if I'm not wrong, it works only if you buy it properly.
I never used it. Actually, long back, somewhere when I was a PhD student, some almost 7, 8 years before I used. After that, I never used. So I
I
don't.
mean, Mendeley can also be used. I've used Mendeley, and it's pretty easy. Once you use one kind of reference setting software, the others also goes almost along the same lines. Zotero is one of the latest ones which many people are using.
Okay.
Yeah, I can share you a link. Recently I came across a kind of lecture on different citing softwares. I can share you the link in the group if that's
Okay. Okay. Thank you.
okay. Pranita, you were asking something.
Yeah. Thank you so much. So I was asking, what is the minimum number of studies that we must have for uh,
See,
performing a
the
meta-analysis?
res- the no the research question itself, when you formulate it properly, that automatically cuts it down to almost like less than 100 papers, if I'm not wrong. Okay, if you see any kind of big publications of systematic meta-analysis reviews in big publications like Nature and Science also, you see that it is almost less than 50 if you see. So the question, the procedure itself filter out and puts you with a very limited number of papers. Yes, less than 100, less than 50 more. As I told you, it can be as less as 5 to 7 also, or as high as it should not be more than 100, because it will be very difficult for you to look into that data and formulate properly.
Right, right. So,
Hmm.
is it like this that if we are taking large number of studies, like 100 studies, that meta-analysis will be a better one, or if we are taking 25 studies or 30
No,
studies,
no, nothing with the
it,
numbers. Nothing with the numbers. It is mostly about the
nothing with the
question you're trying to answer. That is what will be the strength of your systematic meta-analysis, not the numbers there.
Okay, okay. Thank you.
Okay. Any other question, Rakesh and
Ma'am, I have a
Violet?
related
Yeah.
question to the previous one. Ma'am,
Mm-hmm.
what is the maximum number, means the standard papers that we can take? That is you told that there is no such minimum numbers. Okay, we can take, means we can narrow down, and whatever the number we get, that is okay. But what happened for the maximum number? Means what is the standard for that?
See, that's what I'm saying. 1,000 can't be a maximum numbers. Even if you start, see, a simple Google search, say, suppose if you keep on narrowing or adding key terms properly, you'll get hardly 100 research, outcomes there. So I'm pretty sure it's not never go into thousands. Like, if it is a broader question only, and that also if you get proper number of results where it is similar, or in all those 1,000 studies, yes, you can do it, but it will be extremely laborious. Okay, so there's no such thing as very minimum or maximum. Of course, you can't write to a review or systematic review based on just 2 studies. You'll have to have at least a minimum of 5. So greater than 5, yeah. But maximum, no one can say, or put a pinpoint saying that only this is the number or cutoff. That will depend on your research question you're framing. The exclusion inclusion criteria automatically makes it high or low. But as I told you, it will not be, definitely not be in thousands. That means you're asking a wrong question there. Okay. Any other questions?
Yeah, not related to meta-analysis. You were talking about Mendeley. Even I want to, learn how to use it. So whatever you are having, send it to me also.
I'll put it in the same
How,
group only. Everyone can have access
yeah,
to
yeah,
it. No, that should be an
yeah.
issue.
Because I want to learn something, related to reference management.
Yeah,
So
yeah.
Mendeley, yeah,
Mendeley is there, Zotero is there. And, all these free version also have good number of amount. They give out a space for how many references you get. and Zotero it is 300 MB, but in 300 MB you can put the references of around 8,000 references in that. Same goes with Mendeley, but Mendeley has only 2 GB of space in them. So, but it can take a minimum of 2,000 references in them. But I'll definitely share. So,
Yeah.
Rakesh, you were asking how many search engines I need to be looked for. Sure, as many as possible, as many of them you have access to. As I told you, Web of Science, PubMed. I put in Cochrane library, etc. All these have been there. Okay, looked in enough paper, because every time there is something new, Google Scholar and Scopus. See, Google Scholar problem is Scopus is good enough. It will give a good number of papers. You can even use dimensionality recently. That is also being used even for bibliometric analysis also. Google Scholar problem is when you download it, it will give, there's no limit to that. So you have to spend some time at least to have a exclusional criteria, or a limit. You have to say from 2021 to 2023. If that is the time of year wise limit you put in, yes, Google Scholar is also good enough. Okay, but it is always good to check through other databases also. What will happen, you may not have access to it, but the editor might have access to many good number of things, and then he might suggest this is one particular article you have missed out of it. So that should not be a problem, Rakesh. Hope I'm clear.
Yes, ma'am. Thank you.
Okay, so by 29th, try to finish off this particular session of, two assignments I have given you. Number one, at least check through at least 10 meta-analysis of your interest, and then you create a research question based on which we can go for the meta-analysis or a systematic review analysis. Am I clear here, or? Yeah. So any more questions? If not, we can call it a day, I guess.
If any other questions, please let us know. If there are no questions, I will call it for the day. Thank you, Dr. Manjari, once again for your electrifying talk. I hope all the participants got benefit from this. If you have any questions, you can contact Dr. Manjari through the mail ID. Okay.
Yeah, mail ID or in the WhatsApp group, you can just post in any questions or any kind of discussions you can have. Yeah.
Yeah, please post any questions related to this particular topic to Dr. Manjari. She's a good listener and she'll get back to you at the earliest.
What about the
Okay, if there are
hands-on?
no questions,
This is the part of the hands-on only. First part is you have to do the search. You have to come up with a research question, right? I can't
Right.
teach you that. I've already told you
Yeah.
what are the conditions you have to go through. So this is the hands-on. So the rest of the time, you please search through and come try to look 10 of those research meta-analysis, and you come up with your own interest of meta-analysis. I mean systematic review on meta-analysis. Okay. Yes.
okay. I think there are no questions. Thank you once again, Dr. Manjari, for your valuable time and energy. Thank you. Guys, we are
Thank you.
calling it for the day.
Yeah. Bye all.
Manjari, I think after this, Indrani's talk is there, right?
No. Indrani will taking on 29th, because this will be extension of that. So how do you do that meta-analysis part of analysis, that will be taken up in there. So by that time, these people have to come up with the research questions there.
Yeah, noted, Dr. Manjari. Thank you. Thank you
Okay.
all. Yeah, I'm calling it for the day.
Thank you.
Bye. Good night.
Good night all. Bye.